Hello, everyone. Thank you for joining this installment of Boyce Thompson Institute's Breaking Ground discussion series. My name is AJ Bushy, and I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome Aleksandra Skirycz, a biochemist here at BTI who specializes in uncovering the functions of small molecule metabolites in living organisms. And today we'll be talking about her research into using a technology called mass spectrometry to discover how metabolites interact with proteins with the hopes of making discoveries that can benefit plant and animal health. Hi, Ola.
Hey, Jay.
And before we get started with the discussion, a few housekeeping announcements. If you joined us before, you probably know we do like a half-hour discussion between Ola and me, and then we'll do a half-hour Q&A, and you can add your questions in a little chat box. But all the attendees will be muted, and then we'll do the Q&A at the end. And I've also enabled a live transcript button down at the bottom, and you'll probably be seeing subtitles. And if you want to hide that, you can just go into the little CC live transcript and click hide subtitle, and then you won't have to see those anymore. And but first of all, thanks everyone out there for joining us. Looks like we've got a good amount of people, and I'm looking forward to a nice lively discussion. So Ola, let's start with you. Could you tell us a little bit about yourself and how you got into the research you're currently doing?
Yes, certainly, AJ. So when I was preparing kind of like the first slide with my scientific background, I realized that so far I've been a bit of a scientific nomad. And it's because like I lived in many countries and I did really a lot of different kind of research, was involved in all kind of different projects. So I graduated with my master's degree from University of Wrocław, and this is in Poland. I moved to Germany to do my PhD at the Max Planck Institute. And then I did my first postdoc, a long postdoc in Belgium, in a Plant System Biology Institute in Ghent, and I will tell you more about that. And I had a gap year. It was unintended, but it happened. From Poland, I moved to Italy, to Milano, fantastic place, absolutely my favorite place in the whole world.
Very pretty.
And then, yes, and then from, clearly I have the same, taste as George Clooney. And then from Italy, I moved to Brazil. I worked for a company for almost 2 years. And when I was in Brazil, I applied and I got a position in Germany at the Max Planck, first as a project leader and then as a group leader. And from Germany, I mean, obviously now I'm in US, right? So now I'm faculty at Boyce Thompson. And let's see whether it's my last destination. There is still something on the cards here. Mm-hmm. So I think we can move with the next slide. So AJ asked me to pick some highlights. So clearly, one of the highlights was my postdoc in Belgium. The institute, absolutely great place for science, really famous in the plant world for their work on cell cycle and plant growth. And also, Belgium is a fantastic country to live in. Great people, great food, not so good weather, but I mean, you can survive that. And again, it's this old medieval town. It's a real fairy tale town. And also the project was I really enjoyed this project. So I was working in a big group led by Professor Dirk Inze. He's also director of PSB. Mm-hmm. And the goal of the group was to understand better how plants regulate their growth. And the specific aspect I was working on was how plants regulate their growth in response to drought, which is, clearly it's an important thing when it comes to agriculture. And what we discovered, as I'm sure many people before us, and what people are like the newcomers to the field, that plants are very clever in a way they regulate their growth in response to stress. So when you expose plant to a stress, it stops growing, but it's not because it lacks resources. I mean, it has the resources, but it actively decides not to grow to save those resources because plants, as any other organism, they hate to run into starvation. So this is a very, very rapid and very regulated process. And I was particularly interested in the molecular pathways that govern this growth inhibition and subsequently also growth recovery, right? Because if the stress goes away or the stress becomes constant, but it's not threatening, the plant can also recover the growth. And I was focusing on the proliferating leaf primordia. So this is like what I depicted on this 1 cent coin. I mean, this, like this is a small coin, like for people that don't know the euro coins, right? And so this is
about the size of a
penny,
penny, right?
Penny. So this is the kind of organ I had to deal with. So at the beginning, the leaves, when they proliferate, I mean, this is when the cells divide. They're really tiny, and so I spent hours and hours under the microscope extracting those miniature leaves. So this was maybe not the highlight, but you have to do what you have to do, right? Was the highlight? The other highlight was being involved in a fantastic team and building a phenotyping. This is what we call a phenotyping platform, and this is a kind of a device that allows us to study plant growth under different conditions, and it would include watering station and a photographing station, and it was fun because this was really built by the team in the institute. So this is something that we designed, and then we found a company to get all the parts and then and program the whole thing. It misbehaved for a long time, and it often was a real carnage. But in the end, it did work. So this was very satisfying.
So that kind of like black belt thing, that's a camera and it moves around taking pictures, basically.
Exactly. So here the pods were static, right? And then there was an automatic arm. So when I arrived first in Belgium, I was the automatic arm, but then we got the robot doing the job, and this arm would be picking each pod and then taking the pod to the different stations to weigh, to water, to photograph.
Oh, I see. So the camera is static.
The camera is static.
It's like I see like a zoom lens coming down. That's interesting. So that will probably be kind of similar to what we're building here at BTI now, the phenotyping facility.
I think Magda has a much grander plan. So what we're building here, will be the deluxe. This was just a basic model, yeah.
Yeah, very cool. So this research, this sounded interesting. You mentioned, when plants undergo stress, they kind of like stash resources to the side and save onto them in case the stress goes away. How did that get you involved in like looking at metabolites? How did that connection happen?
Not much connection in terms of science, the connection in terms of people, because when I was at Ghent, I met my future boss, the boss that hired me in Brazil to work in Brazil, and this is where really my fascination with small molecules started. I mean, saying that, it's not possible not to work with small molecules. So of course, like during my PhD, during my postdoc phase, I work with small molecules. I work with plant hormones, but this was not really, at the center of my research. So now we can switch slide to Brazil. So it was also, Ghent is a great place. Brazil is also a great country to live in. Again, fantastic people, beautiful country, great weather, unlike Ghent. And I was working in Belém. Belém is an Amazonian city. It's a coastal city. It's a metropole. It's 2 million people. And one of the focal points in Belém, like in Ithaca, is the farmers market. It's slightly bigger than the one in Ithaca. And at least for a European, it's also slightly more exotic, right? So one of the things you could buy at the market in Belém were herbal remedies. Really, a lot of stands with all kind of medicinal plants and concoctions. I never had the courage to try too many, but clearly they have fantastic properties. Otherwise, they would not be so popular. And there is a large population of people in Brazil that still rely on this herbal medicines. Mm-hmm. And in fact, when you think about Amazon, when you think about Brazil, right, you think about biodiversity, and an angle to that would be also chemical biodiversity that exists. And so the term bioprospecting, it refers to harnessing this chemical diversity present in living organisms to develop medicinal compounds to extract, and then of course those can become drugs. And if this is done kind of in a clever way and there is a proper regulation around it, it can actually fuel also biodiversity protection. So on one hand, you harness biodiversity, so you can also see it as exploitation. But if this is done, like I said, cleverly and responsibly, part of the profit can also go into conservation.
That makes sense.
And that makes sense. And of course, there is a lot of efforts, including in Brazil, to really tap into this chemical diversity of compounds. And here I'm just showing one example. This is a small shrub called jaborandi. This is a native name for that. And this shrub is a source of anti-glaucoma drug. It's an actually FDA-approved drug, and it's native communities. They knew about those medicinal properties for a long time, and then they were kind of first discovered by the Westerners in already 19th century. And then the compound that is responsible for those properties, pilocarpine, was extracted, and is also a basis for the drug. So the properties, they don't sound very appealing because it basically causes excessive sweating and salivation. But for some, in some instances, it's actually desirable. And it's also a very nice example to use because who produces this drug is Merck, and Merck, the supply of the plant comes from the like the people that gather that would be coming from the native communities, and it's quite well organized. So they also get fair price for the product they supply. Unfortunately, we don't know how to synthesize or make this compound for organic synthesis. And also, unfortunately, when people try to grow this plant in an agricultural setting, they manage, but the yield of the compound is very low. So it shows that, to get a good yield of the compound, you really need some kind of environmental condition. And
so it only makes the compound when it grows in nature. That's interesting.
Basically, it always makes the compound, but to get like a good yield that actually, makes it profitable to extract, the plants has to be harvested from the natural ecosystem. And because, this is a shrub, it's also important not to over-harvest, right? So it has to be done in a responsible manner, otherwise you will exhaust the natural population. So this is just an example of one plant, but being in Brazil and reading about chemical diversity and how amazing small molecules are, this is what really got me into small molecules.
That's fascinating.
That's Brazil.
And how do you want to talk a little bit about how plants make the compounds? Okay, here we go.
Here we go. So this is really to bring, like switch gear to what I do now. So this is what I started, in Germany, like the whole small molecule exploration project. And as I mentioned, we think, I hope some of my group members also, that metabolites are fascinating. And I mentioned already that there is like this huge chemical diversity of compounds, and we don't even know how many compounds organisms make, how big is this chemical diversity. So like only in plants, it's estimated, from all the way from 100,000 to 1 million compounds. So many of those compounds are still to be discovered, so of unknown nature. But what also, what kind of for me was intriguing is that for a lot of compounds that we know, including what you would think fairly well characterized compounds, we don't really understand their function. And one compound can have multiple functionalities. And also there are compounds, and this is just an example here, like dopamine, that we fairly well characterize what dopamine does in animals. But dopamine is also produced by plants. And what it will be doing with plants, we don't really understand. I mean, clearly plants don't synthesize dopamine to please us, right? I mean,
right.
they do it for themselves. So, and this is really, I have to say, that is the understanding the function of small molecules that lies at the heart of my research.
So how do you discover the functions of these molecules once you find them, and once you find the million compounds? How do you figure out what they do and figure out which ones could be valuable?
So this is the next slide, and here, as usual, there is not one way, but you can do it. You can approach the problem in multiple ways. Mm-hmm. What we decided to do, and this was when I say we, so it was not only me at the time. So it was a discussion between me, my fantastic boss, and the Max Planck Director of Emitter, and some of the people in the group. We thought, okay, small molecule as we know it, they would most often function by interacting with proteins, and proteins are also, in contrast to small molecules, which are small. Proteins are the kind of macromolecules, and they are, you could call them like a muscle. A lot of things that are executed in the cells is done by proteins. So small molecules would interact with proteins, and then they would modulate the function of proteins, and this is how they do what they do. And we also clearly know much more about the different roles of proteins that we know about small molecules. So we thought one way to understand the role of a metabolite is to find its protein interactors. And this is really kind of, you can go to this proverbial, show me your friends and I will tell you who you are. So this is really building kind of a social network of a small molecule to understand what it's about.
So is this kind of an example of how people have been studying like genes and proteins for a long time, and studying metabolomics is relatively new? Is that a big reason why there are so many
questions about the roles of small molecules?
Yeah.
Yeah, I mean, like methodology and like accessibility of certain things is always a thing. I guess the diversity, the chemical diversity of small molecules also plays a role. But it's good because then I can, I found a niche, right? So there are plenty of things to be discovered.
Right. So what are you doing in your lab these days?
So if you can switch the slide. And this is, this is basically past, current, future. So this is really, my group in Germany. It all started in 2015, the whole adventure. So clearly, when you start a new topic, a new group, you need tools, right? You need methods. And for us to study protein metabolite interactions, we needed a machinery to somehow separate protein metabolite complexes, and this is often done using FPLC. It's a picture. It's this kind of a first piece of equipment, and then you also need a way to measure metabolites and to measure proteins, and this is done using mass spectrometers, and this is what you mentioned. So this is our toolbox. It's an expensive toolbox, so thank you, Max Planck, and thank you, Boris Thompson, for being so generous with us. So once you have your toolbox, your equipment, your methods, obviously one also decides for a model organism. And for me, historically, so like I'm a plant biologist. My favorite plant, Arabidopsis. But plants are complicated beings. So sometimes, especially when you establish things, you want a simpler model. And we decided in my group for a unicellular organism for budding yeast, or Cerevisia. And Cerevisia has many advantages. It's also super interesting because it's very important for biotechnology. Okay, so we have
many things are produced in yeast, right?
Many things are produced in yeast, yeah. Including some beverages, but Yes, yeast is good. And, like the way there is also certain aspects of yeast metabolites that are relevant for example, for cancer. So the way they regulate and they govern their metabolism. So it has, this extra interest. And one of the things my group is also interested when it comes to biology is regulation of metabolism. So this is those will be the model organisms, and then the kind of the first output of our product of our work would be what we call protein metabolite interaction networks. And here, all the nodes, so triangles and circles would be either proteins or metabolites, and then all the edges would be interactions. And those are quite nice to look at. But they also contain, a lot of interesting stuff, those networks. They're often very complicated because a single metabolite can interact with multiple proteins, and then the proteins can also interact with each other. And then like kind of the big task is to use those a bit, like you would use a map. And I purposely put this not so maybe detailed and correct map, because often those networks, like we are also at the beginning, right? It's not always 100% accurate, but
right.
Kind of it's enough for us to scout the territory, right? To try to navigate. And then we use those to find interesting molecules, to find interesting interactions. And as with I mentioned treasure hunt, right? So you hope for a good thing, and I exemplify it with my. It's a kind of a tribute to New York and one of my favorite TV characters. So sometimes you get a Manolo Blahnik. I've never seen a Manolo Blahnik in my life, but I imagine it's something wonderful. This would be the treasure you want, but sometimes, of course, you don't find something exactly what you want, right? And I think we can move. So this is basically, like in a nutshell, what my group does. So AJ, you asked me also to tell kind of a bit about the methods we are using, right?
Right.
And I will try to make it kind of keep it simple. So imagine that like the top graphic. So those would be complexes, right? You have proteins and you have small molecules, and some of them happen to interact with each other. So how can you figure out who interacts with who? So you can do it, what people call often biochemical fishing. So, and here a bait would be either your favorite small molecule or your favorite protein, and you use this molecule as a bait, and then you pull, and you hope that what you pull is actually worth something, yeah? And sometimes it is, yeah? And this is how you reconstruct. But you can imagine that this like considering that you have tens of thousands of proteins and even more small molecules, right. Like to do it systematically, it would be extremely laborious, and for certain like beyond the capacities of a single group. And also when it comes to small molecules, like when you pull, you have to know the identity of the small molecule. If you don't know the identity, the chemical identity, you cannot do that. So this was kind of frustrating for us, and we thought, how can we do it in a completely untargeted manner, so without having to have this bait? Mm-hmm. And we decided to go back to a very classical biochemistry and try to separate complexes also from single proteins, from single metabolites. So basically, you have a certain, you can manage this like a jigsaw, and then you break jigsaw to single pieces, and then you look at each piece separately, and then you try to put it back together. So, for example, one property that distinguishes between different complexes and molecules is size, right? So the other property would be polarity or charge, right? So we can all we can use it to break to small pieces and then investigate piece by piece.
So it's almost like you're taking a big jigsaw and not breaking in as like back down to individual pieces, but kind of chunks of like, here's a corner of 20 pieces, here's a chunk of like 10 pieces, and then and then analyzing each of those chunks is kind of what it sounds like to me.
Yes, that is a good analogy. That's what we do. This is why sometimes we have to have, like a different ways to break it, and only by combining the different ways to break it, then we can figure out what the picture was. And it's still often very fuzzy, yeah. But it's a good starting point, as I said to you.
Yeah. So could you give us an example of an interesting result that you've come up with using this PROMIS method?
Yeah, sure. So this is an example of of a molecule that one can say that started like in this old bad shoe, but with enough love, you can turn it into a manolo-blahnik. So the compounds that really caught our attentions are called dipeptides, and they are protein degradation products. And although there are reports that those compounds have interesting bioactivities, there was not so much research. Because they are protein degradation products, right? So probably a lot of people thought, okay, that is kind of a junk, right? I mean,
right.
Protein degrades to dipeptides degrade to amino acid, that's it. We were not so kind of like omnipotent at knowing, oh no, this is clearly not the thing. We only got interested in those compounds because we found them together with proteins, and not any proteins, but really super interesting proteins. So proteins involved in metabolism. So, in metabolism is basically like how, without metabolism, you cannot imagine life. This is how we intercovert compounds between each other. This is how we produce energy, right? This is how we produce building blocks. So this is why we decided to focus on dipeptides. It's a big family of compounds. It's 400 compounds, so we could not do 400. But based on we had a different criteria, and we selected a couple of them to further investigate. And this is just an example of an interaction network. So if we move to the next slide. So what we found, kind of looking by in detail characterization of this particular compound, is that it changes the way that it changes basically and I will try to use not the complete. Okay, so it changes the way. So when you have carbon, right, in the cell, you have glucose. So it changes to where this glucose goes. So, this leads to the accumulation of the production of a compound that is a redox compound that is very important for stress responses. Mm. And in that way, when you treat with this particular molecule, it actually makes plant more resistant to oxidative stress, which is fantastic. And recently, we also found that it also prolongs the lifespan of C. elegans. And this may be not surprising because those compounds are very evolutionarily conserved, and also primary metabolism or central carbon metabolism is also a very conserved evolutionary feature.
Yeah, it's kind of interesting. So these are the products of biodegradation. So proteins are breaking down in the cell. So that kind of makes sense. That might be a sign of stress, and then this dipeptide kind of would be like a signal for the rest of the cell to, hey, batten down the hatches, we're under stress here, kind of. That kind of makes sense to me intuitively. That's interesting.
This is what we think. So, and I mentioned to you that sometimes those networks are pretty complicated. So we to get to this kind of interaction network, I mean, it takes a lot of work, and it's also expensive, but you can do it, right? And then the way it's really challenging, because you cannot do it en masse. You have to do it one by one. It's in detailed characterization of the interaction. Okay, your small molecule interacts with this protein. Okay, you have to validate the interactions, and then you have to figure out what this interaction is good for, and whether this is actually relevant for biology. So. Initially we focus on those interactions between our compounds and enzymes, but we know that there is more to it. So if I can have the animation. So brace yourself, there is more coming because now we have other interactors for this particular small molecule. So we think that it does more than just regulate metabolism, but I hope that I can update you maybe in a year time in the next breaking ground.
Yeah. Take you to the upside down. Is that what it's called?
Absolutely.
I think we have time for one more. If you have another example you can tell us about, and if folks have any questions, you could start putting them in the chat box, and then we could do a Q&A after this one.
So this is an example of yet another degradation product, but degradation product of nucleic acid. And again, this compound was like, I mean, people knew about those compounds, but they were not so much interested in studying their biological function. And again, we decided that they are exciting because we found them together with proteins, and especially with proteins that form a non-membrane compartments. This is something I will not go to, but basically it's what those compartments are is little stores. So when plants experience stress, and not only plants, also other organisms, they have certain proteins or certain transcripts that they don't need at the time, right? If anything, maybe they actually don't want them. So for instance, I told you that plants inhibit growth when they are exposed to stress, right?
Right.
So maybe at this particular time, they don't want to have those growth regulators present and active. But they have them, right? And then invested energy to make them. So to break them is a bit wasteful. So what they do is they create some kind of like a small pantries or like storage units to keep those things aside within those compartments. Those proteins cannot work, but they're happy kind of there, right? And then when the stress ceases, you can very rapidly bring them back to action. And we figured out that this compound, our compound, is involved in the formation of those fossils, of those storage fossils. And again, it's an example of something that is very evolutionarily conserved, both on the side of a molecule and on the side of this particular process. And also, it's like I'm very excited because this compound also, and maybe not so much the compound, but those fossils, they bring together me, Magda, and Andrew, because in addition to proteins and small molecules, they also store RNAs, and this is Andrew's favorite thing. And we think that they are involved for regulation of plant growth, and this is what Magda is interested in. So
yeah. So have you this is all in Arabidopsis, is that what you said? Have you?
Yes.
Have you looked for this in other plant species, just out of curiosity, to see how conserved it is over? Like...
not me, but I mean, for the compound, I don't know actually. Like we never looked for it's an evolutionarily conserved compound, but we never studied the role in different organisms, but Arabidopsis. But those foci, they are studied, across organisms. So that's kind of one of those hot topics nowadays in biology.
Very cool. Okay, well, that's fantastic. I think we have a couple more slides, and then we'll switch to the Q&A.
This is really to say thank you to people. So like molecules, we don't live in a limbo, right? We interact. Sometimes in a good way, sometimes not so good way, but I mean, my interactions with, I have to thank you here, like for people for many wonderful interactions. So this is my group in Germany, and the big picture of this man there is Lothar Wilmitzer. He was my director and mentor and a friend, and he supported me in this project. And he retired actually last week, and I retired with him from the Max Planck Society. So now all my heart and body belongs to Boyce Thompson.
Yeah,
and then
we have you full time now, right?
And then the people on the picture, I mean, besides one guy, Dennis, that is finishing his PhD, they also already, they found new positions. So, for instance, Monica, who was my postdoc, she's now her own boss. So she has a group in cows. She's a professor there, continuing on the small molecule research. And then also my fantastic group at Boyce Thompson, Romina, Mani, Venkatesh, and Mateusz. Mateusz was the first person that came with me and helped to establish the lab. And then Venkatesh really took over, and he's now in charge of proteomics here at Boyce Thompson. Besides looking at the stranger roles of dipeptides. And then Romina is also working with dipeptides and the role in regulation of metabolism. And Mani is a joint postdoc, and she works with stress granules. So this is the project between me, Andrew, and Magda.
Perfect.
And then the next slide. And those will be all like my wonderful collaborators from Germany, but also from Boyce Thompson. I mean, Andrew and Anna, fantastic. I don't know how I'd manage without them. I mean, they also read all the things I write in Sweat, all my grant proposals here. And then Annette, so she was the first person to welcome me. She actually visited me, also my lab in Germany. And we do a lot of stuff with for instance, with Frank's group and with Andreas. And yeah, so really, I mean, what can I say? It's fantastic to be part of a good interaction network.
I like that way of thinking about it. All the different interactions and collaborations is kind of similar to proteins and metabolites, right? All right, well, fantastic. Thank you so much, Ola. This was really fascinating. It isn't a side of plants that I often think about. I'm guessing most people don't think about. Okay, so let's do some questions. We have one question in the chat room from Stephen Rossthorn. If TIR-ASP, is that how you pronounce it, the dipeptide? If TIR-ASP specifically interacts with a suite of protein targets and can change metabolism, how specific is the TIR-ASP motif amongst proteins as predicted from known sequence information?
It's a great question, and you would think, like for if you have many interactors, maybe they would have like a common domain that they bind to. But this is not some like for the suite of those interactors that I showed for TIR-ASP. There doesn't seem to be like a common binding domain. So, but I have to also say that, I put the edge as an interaction, but not all the interactions would be direct binding. Some of those would be indirect. Mm-hmm. And this is, something that also becomes like apparent now. There is more and more people studying protein metabolic interactions on more kind of a system level or protein metabolome level. Is that there is not, one small molecule, one protein partner. There is also not one binding domain and one small molecule. So there can be many small molecules binding to a given domain, and vice versa. There can be a small molecule that binds to different proteins using different binding motifs.
Makes sense. And I would imagine different parts of the proteins as well. Like, most enzymes have the active site, but then there's also allosteric sites that can change conformations and things. Maybe that's something that's going on. Does anyone else have any questions there in Zoom land? I was thinking another one. If you talked about the stress granules, it seemed to kind of tie back to some of the first things you were doing in Belgium. Is this like a circular thing for you? Do you feel like you've gone back to the beginning, but with a new
Yeah. I mean,
how is it?
Yeah, kind of clearly. Sometimes, something falls into your hands that you don't. So when I was in Belgium, like it was really frustrating, right? Because we kind of knew that plants rapidly arrest growth, and we knew some of the players, but we could not completely figure out, how is that happening. And then I left, and I haven't thought much about it later. So, and then we had the stress granule project with my, Monica, who is now in Chaos, was really in charge of that. So she was the lady in charge. And we are biochemistry labs. Sometimes we do this like side projects just because we have the expertise, what we call kind of bread and butter projects. And she thought, Ola, what about isolating those stress granules and seeing what is inside? And what was inside was exactly the growth regulators that were puzzling me when I was a postdoc. Like, how are they regulated? So, without looking for something, sometimes you find it. And that's great. That's great. Yeah.
That's how science goes, right?
Sometimes. That's how science. Sometimes that's how life goes, right?
Yeah. That is true. It's right there. So we have a few more questions. Is it possible or even conceivable to identify the genes or even base pair sequences that code for some of these molecules so that they can be incorporated in other organisms? Because they're metabolites, right? There's not.
Oh, can you find biosynthetic pathways?
Yeah, that's how I interpret that for genes. Could you like figure out the pathway and then put in organism to create these small molecules, these metabolites?
I imagine this can be done for the molecules I work with. It's unnecessary because they are produced by across the board. But I mean, clearly, when you think about biotechnology, you have tons of examples where you transfer the biosynthetic pathway to produce, let's say, a plant compound in yeast, so artemisinin, that is an it's used for malaria, right? Mm-hmm. And you can actually express the pathway in yeast and purify the compound. So in the place I was before, in the Max Planck, so there was one group, actually, it was not in a group, a department, and they had a lot of those projects where they would try to produce certain valuable compounds in not even in yeast, but in plastids, so because plastids are fantastic machineries to produce things. So for instance, they showed that they can produce artemisinin in tobacco plastids.
Wow.
And also the compound, actually, the name, I think it's called astaxanthin, is the thing that salmon eats, that we think that salmon is pink, right? I mean, I think maybe sometimes it's pink, but it needs this this compound, this carotenoid compound. And this was also, they shown that this compound is quite expensive, and it's purchased by the salmon farms to give this color of the meat. Yeah. And this can also be produced in tobacco. I mean, those plants are not very happy, but they produce the compound too.
I knew they were producing, I knew some folks were producing proteins in like genetically modified tobacco. I didn't realize it was in plastids though. Is there a reason to do it in plastids and not like the nuclear genome, or is?
I think, like okay, now, oh my God.
We're getting beyond.
I hope that the people don't watch me do that. Yeah.
You don't have to answer that if that's not really a metabolite question. We have another one. So to the tier asp motif, if it is a breakdown product, are there many proteins that contain this pair of amino acids? And could this relate to specific actions from degradation of these proteins?
And this is again a kind of a great question. What drives specificity of those peptides, right? So when you think just about proteins, like most of the proteins would have tier asp in their sequence. Mm-hmm. So we think that this is something that people often ask, and we try to do it kind of in a simplistic manner and just to see whether certain peptides are enriched in proteins, and they are. But then when you think about the amount, and I mean it matters like how abundant is certain protein, right, or like how fastly it's degraded, then to actually contribute to a pool of a certain dipeptide. Mm-hmm. And probably a lot of that is also regulated at the level of degradation, right? So like not only the enzyme that would cut proteins to dipeptides, but also the enzyme that then would convert dipeptides into amino acids. But yeah, that's one of the, that's one also of the kind of on the challenges on the list to understand what drives specificity of accumulation, why photosynthesis accumulates under certain conditions and not others.
Yeah. That's really interesting. Sounds like there's a lot of cool questions that could still be asked and studied. Yeah.
Always.
Always, yeah. That's a good thing.
That's a good thing.
I don't see any other questions in the chat box. If anybody has another one, now's your chance. But those are good ones. The ones we got were excellent. Okay, everyone out there. Oh, very nice talk. Thank you, Andrew.
Thank you, Andrew.
Here. Well, thank you again, Dr. Skirycz, for taking the time to chat with us today. And thank you everyone out there who joined us for this virtual chat. And please join us next time for our BTI's next Breaking Ground discussion series on Tuesday, April 19. We'll be with Dr. Surya Saha, a bioinformaticist, who will be talking about his work developing solutions for citrus greening disease powered by the next generation of scientists. So that will be that'll be a neat one. So please join us for that. There's some neat, like mentoring aspects of the things he's doing with undergraduates. And you can please go to btiscience.org to get more information and to register. And I'm just going to throw a bunch of URLs in the chat that I will be saying, and that's the first one. And you can read more about BTI's current research and many other neat stories about BTI science in our annual report, which you can find online at btiscience.org/annual report. And thanks again for joining us. BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give, or email our development team at development@btiscience.org. And thank you all out there for your interest and support of the Boyce Thompson Institute, and have a wonderful day and be well. Thanks again for joining us, and thank you again, Ola. Bye, everybody.